Good evening, students. Today, in an hour, I'm going to discuss all the important aspects of evolution in the form of bits. Okay, so please write it down wherever it is necessary for you that you don't know those points you can write down. So we shall start with origin of life on Earth. Before that, we should discuss something about the origin of Earth. Someone has come. Okay. Scientists say that Earth originated 4.5 billion years ago. The origin of Earth is 4.5 billion years ago. After 1 billion years, that means around 3.5 billion years ago, first life originated. And this life is nothing but prokaryotic life. Again, after 1 billion years, in 2.5 billion years ago, prokaryotes evolved to form eukaryotes. So this is the chronological order given by the scientists. Origin of Earth 4.5 billion years ago. Origin of life 3.5 billion years ago. And the first life that is originated is a prokaryotic life. And after that the prokaryotes evolved to form eukaryotes around 2.5 billion years ago. There are so many theories with respect to the origin of life. Some of the early theories are: first one, special creation theory. It is proposed by a bishop or church father, we can call him as his father Sarich. According to this, the entire earth is created by God in 7 days, as per the view given in the Bible. That is what simply is. What you have to remember here is who proposed the special creation theory? Father Sarich. Next one is said to be cosmozoic theory. According to cosmozoic theory, life did not arise on our planet. Rather, life was descended from some other planet where life is already existing there. Okay, due to some meteorites or other some things, life from one planet came to our planet. That means life. So it means life originated from the universe, cosmos, in another planet, not directly on our planet. So, according to this does not explain the origin of life. Rather, it shows that it explains the existence of life. Already there, from there life came here. That means life did not origin. How life originates, this theory does not explain. And the next one is spontaneous generation theory. So, this spontaneous generation theory is proposed by Aristotle. This is also called as theory of abiogenesis. That means life originates from non-life, non-living matter. According to Aristotle, he suggested that fishes and frogs arose from moist soil. That's what he has seen. His assumption or presumption is maggots, the larvae of houseflies, and certain worms like earthworms, etc. They can arise from decaying organic meat, and mice in the field arise from the mud of Nile River. So all these things, these are said to be non-living matter from which the life originated according to spontaneous generation theory. And it was said to be ruled out by the experiments of three different scientists: Marcus Pelagini, Francisco Redi, and Louis Pasteur, who proposed that life originates only from life. Genesis means formation of life. From where? From pre-existing life. According to biogenesis theory, life originates from pre-existing life. They have conducted certain experiments. You can go through in the Google. One experiment is Reedy's experiment. He has taken different containers. He placed organic meat in all these containers. He opened it one, covered it with cork, and covered it with parchment paper or gauze. And he observed that in the one which is open, the maggots are formed and they gave rise to houseflies. And the other two, there is no life originating. That means unless otherwise the flies go inside and what we call lay their eggs, which become larvae and then houseflies, life will not originate. So according to these experiments, spontaneous generation theory was set back. It was ruled out, and they suggested that life can originate only from life. Another one that is called as Spelangeni. Spelangeni also made the same type of experiment, but he used some what we call some gravy broth. We call it as one is boiled, the other is said to be not boiled, and the one which is said to be flask is not sealed. There microorganisms appeared, and the one which is sealed, microorganisms did not appear. This is another experiment. And Louis Pasteur, it is famously called as swan-necked experiment. Okay, swan-necked flask experiment. This you have to remember. So what is the experiment of Louis Pasteur? It is swan-necked flask experiment. So what he has done? He has taken again some broth, heated it so that no microorganism or anything after heating it dies. So it is free of any organisms. Later it was said to be cooled. Now he drew the nozzle into this shape so that after some time he has observed that no life originated here. And when he cut open the neck of the flask, he could see the growth of the microorganisms. So what he has observed in this is some of the microorganisms are said to be uh, present here in the bending portion of the neck. That means they could not travel through and they got stuck here. For example, a housefly entered, but it could not cross through the bend and it was stopped there and it laid it eggs. Something like that happened. So according to this, he suggested again that life can originate only from pre-existing life. Later came modern theory of origin of life proposed by Haeckel, Oparin, and Haldane. But among these three, the one that is given credit is Oparin. And this theory is called as theory of chemical origin of life. Theory of chemical origin of life. Who proposed it? We can simply say A.I. Oparin proposed it. So along with his name, you can add Oparin and Haldane. So it depends on the book from which the question comes. In some books it is only A. Oparin. In some books it is A. Oparin and Haldane. And in some it is written that Haeckel is also added. So based on the question that is given, you can answer accordingly. Haeckel first of all proposed that the most primitive organisms were generated spontaneously from some inorganic matter. So that is what the Haeckel's hypothesis is. So life originated from non-life only according to him. It is the theory of abiogenesis, isn't it? So here, for this inorganic matter to form into organic matter, that is life, it needs certain energy, and that energy is given by electric charges, UV light, corpuscular radiations of radioactive elements. This is what proposed by Haeckel is. And later came Oparin and Haldane's hypothesis. This is the main crux of the origin of life. According to them, Earth's primitive atmosphere was of reducing type. This you have to write down. What is the nature of the primitive atmosphere? When the Earth originated, the Earth is said to be having an atmosphere which is reducing. What does it mean? That is, it is without oxygen. The atmosphere without oxygen is called as reducing type of atmosphere. So here this theory states that life originated around 4.6 billion years ago or 4,600 million years ago. There are different theories, and you need not bother about these theories. And what the theory says that The age of first life, that is 3.5 million years ago, prokaryotes originated, which gave rise in the course of evolution to eukaryotic life around 2.5 million years ago or 2.5 billion years ago. And what this suggests is, first of all, this includes the formation of the solar system and the primordial atmosphere of Earth, followed by production of polymers, origin of nucleic acids, and certain models of life, primitive models of life, protocell models we call them as. These protocell models are said to be three types: coacervates, microspheres, and proteinoids. So what you have to remember is, what are the different, find the protocell models from among the following. So you have to remember these three names: coacervates, microspheres, and proteinoids. So whatever it may be, this entire theory of biochemical origin of life involves only two steps. One is chemogeny, the other is biogeny. What is chemogeny? Chemogeny, as the name implies, formation of chemicals. Formation of chemicals. After that, what happened? These chemicals organize to form life. Formation of life, which is called as biogeny. So we know that every human being, every animal, every plant is simply made up of chemical substances. So without chemical substances being formed, no life can originate. So he suggested that from the primitive atmosphere, simple molecules like monosaccharides, amino acids, nucleotides, that means nitrogen bases, sugar, phosphate, and fatty acids, glycerol, all the things that are required to form the macromolecules like proteins, carbohydrates, lipids, and nucleic acids are said to be formed. And after that, they organized, these compound substances organized themselves to form a protocell, which led to the evolution of life. And coming to the chemogeny. So scientists say that conditions on the primitive earth were reducing, and this reducing atmosphere contains these five things. Okay, we have to um, write down these things. Primordial atmosphere of earth is dash, it is reducing. That is the first question. Second one is, the primordial atmosphere of earth is composed of dash. One is water in the form of vapor or water, and methane, and ammonia, carbon monoxide, and carbon dioxide. When you observe all these five molecules, what in general these have? They have carbon in general, in these hydrogen in general, oxygen in general, and nitrogen in general. So these are the main four elements that are present in the primitive atmosphere. What for these are required? For example, carbohydrate. Let us say carbon, hydrogen, and oxygen. It comprises of carbon, hydrogen, oxygen, C6H12O6. So, glucose to be formed, we require carbon, hydrogen, and oxygen. Let us say fatty acid, palmitic acid, and so on. Palmitic acid or arachidonic acid, they are having a formula much similar like this: C55H78O21, like this. Here also there are three elements: carbon, hydrogen, and oxygen. When you come back to the amino acids, so amino acid is having four functional groups around the C. That is, on one side there is H, on the other side there is R. R means alkyl group, and the other side there is amino group, and on the other side it has a carboxylic group. So in any case, what does it actually contain? It contains carbon, hydrogen, oxygen, and finally nitrogen. And when you go to DNA, DNA contains nitrogen bases, sugar, and phosphate. So here also there is nitrogen. So these are the main components. We are not talking about phosphate. Why? Because phosphate cannot exist in atmosphere. We are talking about only the atmospheric components, and these components in common they have carbon, hydrogen, oxygen, and nitrogen. So when these chemicals are said to be present in water, they combine and recombine to form simple chemical compounds, which combine again to form complex chemical compounds, which again combine to form monomers, and which again polymerize to form polymers like proteins, carbohydrates, lipids, and nucleic acids. That is how the chemogeny or chemical evolution takes place. Okay, and the primitive atmosphere is said to be having certain features. One is said to be hydrogen is abundant and it is free of oxygen. That means no oxygen. Hence, we call it as reducing atmosphere. Water, carbon dioxide, and carbon monoxide are present, and ozone layers are also absent. Because ozone layer is absent, how ozone is formed? When the oxygen is broken down, it forms oxygen free radicals. So when the oxygen free radicals combine with oxygen to form ozone. So for which oxygen is actually needed. But here we are saying that there is no oxygen in the primordial atmosphere. And there is no oxygen itself, how can ozone be formed? When ozone is not there, what happens? There is no any layer that can cover it. Now the UV rays from the universe directly reach the earth, and this acts as the source of energy for the formation of all these different types of chemical compounds during the process of chemogeny. Okay, and simple organic compounds are formed first, like acetylene, ethylene, methane, like that, and they gave rise to certain hydroxyl derivatives or oxy derivatives like acetaldehyde. When acetylene combines with water, it forms acetaldehyde, and in due course of time, different acetaldehyde molecules combine to form acetaldol, and these get polymerized to form large compounds, and thus led to the evolution of what we origin of chemicals like fatty acids and glycerol. That is what the scientists say, and several of these reactions led to the development of amino acids also. Okay, a glycolic acid is formed, which combined with ammonia to form glycine and releasing water. So different types of all monomers. Carbohydrates, the monomers are monosaccharides, for which monosaccharides have to be formed. Unless otherwise they are formed, polymerization cannot take place and carbohydrates cannot be formed. For proteins, amino acids are required. So unless otherwise amino acids are produced, protein cannot be there. And for fats, triglycerides, fatty acids are required. Unless otherwise the fatty acids are formed, we cannot have triglycerides. So what the scientists say is, during the chemogenic process, simple substances are formed first, followed by polymerization of them to lead to the formation of complex substances. Okay, and all these chemical substances that are formed, organic compounds, they are said to be present in the seawater. So, in the seawater, these chemical substances are present, and this seawater is said to be somewhat like a broth, which is hot and soup-like thing. Because when we go to hotel, we take soup. Soup contains water and different types of chemicals, different types of ingredients. Likewise, here there are so many ingredients here, and Mr. Haldane called it as hot dilute soup. This you have to uh, remember. So, who coined the term hot dilute soup? It is nothing but Haldane. What does hot dilute soup refer to? It refers to the organic compounds that are accumulated in the water body. What is it is also called as? It is also called as hot thin soup or prebiotic soup. And the formation of this hot dilute soup set the stage for various chemical reactions leading to the formation of complex organic compounds like polysaccharides, fats, nucleotides, nucleic acids, polypeptides, etc. Now there comes what we call the energy requirement. The energy resources for all these chemical reactions, for the polymerization reactions, is drawn by electrical discharge as suggested by Haeckel before, lightning, solar energy, ATP and other pyrophosphates that led to the formation of complex organic compounds from simple organic compounds. So thus, it can be simply shown by this diagram. So primitive atmosphere of Earth gave rise to different chemical compounds. One such chemical compound is amino acid. We are taking this as one example. And these amino acids gave rise to proteinoids. That means combination of amino acids gave rise to proteins. Because these proteins are primitive, we don't use the term proteins, rather we use the term proteinoids. And proteinoids in the water combine to form certain protocell models. What are protocell models? The primitive cell models. And these protocell models, according to scientists, are also exhibiting a process called as replication. So replication of protocell models takes place. And in due course of time, nucleic acid synthesis has taken place, and they gave rise to even what we call protoribosomes. What are protoribosomes? Without ribosomes, there is no protein synthesis at all. Scientists say protoribosomes were evolved, and along with this, nucleic acids evolved and genetic coding mechanism also evolved, and that gave rise to the contemporary cells in simple terms. And these protocell models, we call them as protobions. So here, for the formation of protobions, three conditions have to be fulfilled. First one, continuous supply of self-producing molecules called replicators. Why continuous supply of self-reproducing molecules are required? Because let us say there is C, and this a protocell is formed. Okay, primitive cell is formed. This primitive cell has to sustain itself in the water, unless otherwise life cannot originate. For example, human beings are there. How human beings are maintained over generations? Because human beings reproduce, they replicate themselves to form their children. Likewise, this cell protocell should have a should have the capability to produce its own molecule structure. So they must be replicating. Second one is copying of these replicators must have been subject to mutation. So new things have to be formed. Unless otherwise, there is no evolution at all. So these replicators, not only they have to replicate, they must be able to undergo mutation, and for which they require free energy, without which they cannot sustain itself. So these are the three conditions that have to be fulfilled for the formation of protobions. And this concept is proposed by Oparin along with Sidney Hawks. Sidney Fox, sorry. Sidney Fox and Oparin suggested that complex organic compounds synthesized abiogenetically. That means they are formed before the life originated. They are formed from inorganic or organic matter. They are formed from components of the primitive atmosphere. There is no life that origin at that time. So thus these organic compounds are synthesized abiogenetically on the primitive earth. Later they accumulated and formed colloidal cell-like or aggregates. And these cell-like aggregates were called by them as protobions. So you have to remember this. So what are protobions? They are colloidal cell-like aggregates formed by the combination of different complex organic compounds that are formed abiogenetically. Who proposed this? Who coined the term? Oparin and Sidney Fox. And it led to the evolution of first non-cellular forms of life. First non-cellular forms of life. So these non-cellular forms of life are nothing but they are chained molecules containing RNA, protein, carbohydrates, etc. But the scientists say that their main drawback was that they were unable to reproduce. So the first thing that is required is they must be self-replicating. But here they suggested these non-cellular forms are unable to reproduce. So these microscopic, spherical, stable and motile aggregates which are not able to reproduce were called as coacervates by Oparin and as microspheres by Sidney Fox. So these two terms you have to remember. One is coacervate. Who coined it? Oparin. Second one is microspheres. Who coined it? Sidney Fox. What these are? These are nothing but first non-cellular forms of life which cannot reproduce. As these Koya cervix do not have lipid outer membrane and cannot reproduce, they fail to fulfill the requirements as a candidate of probable precursors of life. So, first of all, Koya cervix drops are found within the sea, but they are not the precursors of life. Why? Because one, the first thing is they do not have a lipid membrane around themselves. Second thing is they cannot reproduce. That is the reason why these non-cellular forms of life are not considered as the precursors of life. Why for life lipid is required? Let us say life originated in where? When somebody asks you life originated in dash A water, B land, C air, D all of the above. What you have to mention? It is water. So life originated in water. So in water, if light originates, a protocell is formed with a membrane around it. It contains DNA, RNA, proteins, etc. But this membrane is made up of carbohydrates. What is the property of carbohydrates? The carbohydrates are soluble in water. That means soon it gets dissolved. So there is no protocell at all. Let's say if it is made up of proteins, and we know that proteins are also soluble. Soon there will not be any protein at all. There will not be any cell at all. Let us say if they are said to be surrounded by lipid, we know that lipids are insoluble in water so that they separate the external water from the internal water and thus they can sustain. If these are self-replicating, what happens by processes like budding, fragmentation, binary fission, if they are said to be self-replicating, we can say that life life can originate. So first thing that is required is to be called as a precursor of life. This molecule, this protocell should be surrounded by a lipid membrane. And now we come to the formation of primitive life, which is called as biogeny. And according to this, all these molecules, first of all, coacervates, microspheres, and they gave rise finally to form a first organized cell with a lipid membrane outside containing all the polymers like DNA, RNA, proteins, and result for food in the form of carbohydrates. And this is said to be first prokaryotic cell according to this theory. But scientists say that is it true that life originated from the chemical substances, their polymerization? So there came two scientists, Stanley Miller and Harold Urey. They wanted to prove this abiogenic molecular evolution of life in 1953 when Watson and Crick were proposing the double helical model of DNA. These two were doing an experiment to prove this um, origin of life from chemical substances. So they have experimentally proved. How they proved? They have taken the boiling water, it was sent out, it was condensed, and it was sent through a place which was prone to spark or discharges. So that what actually required here? Life to originate, it requires energy. For energy is to be provided by different sources. What are those sources we have seen previously? The sources are ఎక్కడ ఉంది అది? Electrical energy that is produced during lightning and also during decay of radioactive elements, solar radiations, and volcanic eruptions. These are the sources of um, energy. So the same source is said to be provided in the form of electric current, and this particular bulb is now containing the all the substances that were suggested by scientists to be present in the primordial atmosphere. The primordial atmosphere is found to contain methane, ammonia, water, hydrogen, carbon dioxide, carbon monoxide, light, etc. And the main components here are methane, ammonia, water, and hydrogen. Hydrogen is abundant, we already have seen it. So these were prone to electrical uh, ch- charges. Now what happened is, after that they were allowed to, this water is allowed to carrying all these uh, compounds that are formed due to electrical discharge here, they were allowed to pass through a condenser. So condenser is the one which cools the water that flows through it. So this is said to be cooled and it is collected in a container, and this container is found to contain what we call organic compounds like amino acids, fatty acids, etc. So based on this, they can suggest that life before life originated, first of all, organic compounds originated, and this is followed by cosmogeny, that is nature of primitive life and its evolution. They say that earlier cells were anaerobic. You write down this sequence. Earlier cells were anaerobic. That means they obtained energy by fermentation of some organic molecules as no oxygen was available. Okay, so these anaerobic organisms are said to be nothing but prokaryotes. So these are anaerobic prokaryotic, and they derive energy from organic molecules only. Thus, they are chemoheterotrophic. Heterotrophic means ones which depend for their energy on other sources. They cannot produce energy, so they depend on other sources. And here, what are the sources here? Chemical compounds. For example, we know there are sulfur bacteria, isn't it? For which hydrogen sulfide is broken down to release hydrogen and sulfur, during which energy is released, and that energy is utilized by sulfur bacteria. So here the source is hydrogen sulfides. Thus they are heterotrophic, and this source is a chemical substance. Hence they are called as chemoheterotrophic. So the first organisms are anaerobic, prokaryotic, and heterotrophic organisms. Okay, remember this. These started deriving nutritive organic compounds from the primitive seas as their building blocks and energy sources. So, because they are drawing much energy at the time, there is so much energy, they grew to enormous size. And to restrict their size, they started cell mitosis, and now instead of growing in size, they started increasing in number. This is the first concept that was established during the evolution of life. Anaerobic prokaryote chemoheterotrophic organisms started increasing in size, followed by starting mitosis, leading to increasing the number of these cells. Okay, and as this is happening, they are continuously using all the chemical compounds to derive the energy. Previously, there are only tens of these protocells. Now there are said to be millions of these in the sea, and all these are drawing all the chemical compounds in the sea. Now there is supply of organic compounds is depleted. That means it is gradually coming down, and now they have evolved another mechanism, that is what we call it as chemoautotrophic mechanism. So previously they are anaerobic, prokaryotic, chemoheterotrophic. Now they evolved to form anaerobic, prokaryotic, chemoautotrophs. What about chemoautotrophs? They started synthesizing their own organic food from inorganic compounds in the presence of chemical energy. So they do not require external energy source. Rather, they depend on the sources on their own. And such an examples are present day sulfur reducing bacteria, nitrifying bacteria, iron bacteria. Okay, and how these evolved? They evolved due to mutation. So this is the second criteria that we have discussed previously. So anaerobic prokaryotic chemoheterotrophs gave rise in due course of evolution to anaerobic prokaryotic chemoautotrophs. This sequence we have to remember. Okay, if any question comes, it may come from this part. And the next one is facing the problems of increasing deficiency of inorganic compounds. Now inorganic compounds are depleting because their number is said to be increasing. Some of these chemoautotrophs developed what we call bacterial chlorophyll, not the chlorophyll that is present in the plants. We can call it as chlorobium chlorophyll. Na? So chlorobium chlorophyll like thing started, and they started photosynthesis. Now we call them as anaerobic prokaryotic photoautotrophs, and we can see them in present days in the form of blue-green algae or cyanobacteria, which have chlorophyll in them. And this evolved about 3500 to 3800 million years ago. So, anaerobic prokaryotic chemoheterotrophs gave rise to anaerobic prokaryotic chemoautotrophs, which gave rise to anaerobic prokaryotic autotrophs. What is common among all these? All are prokaryotes only. That is, the first life that originated on Earth is prokaryotic life. So, what happens when photosynthesis takes place? Can anybody tell me what is the reaction of photosynthesis? Photosynthetic reaction. Excitator, huh?
So,
Photosynthesis reaction ఏంటి? Equation. తెలీదా?
but
Sir,
6CO2 plus
6CO2
1H2.
in the presence of sunlight
In the
rise
presence of
to
sunlight. Very good. Next.
P6H12O6 glucose plus oxygen, sir.
Hmm. Complete reaction. What are combined?
Um, CO2
CO2 plus
CO2
H2O.
plus H2O
H2O gives rise to
gives rise to
C6H12O6.
C6H12O6 plus
During this,
6H2.
oxygen is released.
Oxygen
So
ATP, sir. ATP.
during photosynthesis, carbon dioxide is taken to the surface and oxygen is released. So primitive atmosphere suggested by the scientists is reducing. There is no oxygen. Now what happened? They evolved, they started photosynthesis, that they started releasing oxygen, from which previously they are anaerobic, now evolved aerobic photoautotrophs. Okay, they evolved aerobic photoautotrophs. That is what cyanobacteria are. And during the course of time, what happened? Prokaryotes are evolved. Now we have to see the evolution of eukaryotes from the prokaryotes. There is a theory proposed by two scientists. One group of scientists is Raff and Mahler. In 1972, they proposed that first eukaryotes evolved through either mutation in prokaryotes. They evolved through mutation in prokaryotes. So prokaryotes got mutated to produce eukaryotes, according to Raff and Mahler in 1972. Another suggests that eukaryotic cell organelles might have arose due to invagination of the cell membrane. And the third, proposed by Margulis in 1970, proposes that symbiotic association of different prokaryotes led to the origin of eukaryotes. And later, algae, fungi, protozoa, and other living organisms, starting from protozoa to primates, still primates, human beings, the evolution has taken place. So let me explain this part once again. Mm, just a second, I'll take a slide. Okay. So now what happens is, this is prokaryotic cell. Prokaryotic cell, what does it have? It has a cell wall around it and a plasma membrane and a circular DNA, 70S ribosomes. Other than this, there is nothing here. And it has to evolve to form a eukaryotic cell, which has plasma membrane. If it is a plant cell, it will have cell wall. If it is an animal cell, it will not have a cell wall. So I am discarding this thing. So plasma membrane, inside which there is a nuclear membrane, inside which in the cytoplasm there are ribosomes which are ATS type. In addition to this, we have so many things. Mitochondria, we have Golgi complex, we have endoplasmic reticulum rough as well as smooth, we have lysosomes, centrioles, etc. Okay. So what is common among all these cell organelles? Can anybody tell me what is common along all these cell organelles?
Membrane, sir.
Very good, excellent, membrane. There are three types of cell organelles in a eukaryotic cell. First one is cell organelles without membrane. Cell organelles without membrane. Example, can anybody tell me? Ribosome. The cell organelle without any membrane is ribosome. So whatever the ribosome that is present here, it evolved to form this ribosome. No membrane is required here. And second category of cell organelles in eukaryotic cell are cell organelles with single membrane. Cell organelles with single membrane. Can you mention what are these? Cell organelles with single membrane.
Nucleus, sir. Nuclear membrane.
Nuclear membrane is double membrane. Cell organelles with single membrane are endoplasmic reticulum. It is only made up of single membrane, isn't it? Whether it is RE or SER. Golgi complex, cisterna, this is also single membrane. Lysosomes, single membrane. So all these are single membrane cell organelles. And cell organelles with double membrane, these are the third category. That is nucleus,
Nucleus, sir.
mitochondria, and in case of plants, chloroplast. These are the double membrane cell organelles. So if you don't know anything about this, please write it down. Cell organelles in eukaryotes based on the membrane are of three types. Cell organelles without membrane, that is exemplified by ribosomes. Cell organelles which are having single membrane, endoplasmic reticulum, Golgi complex, lysosomes, etc. And cell organelles with double membrane. If we consider nucleus also as a cell organelle, there is a nucleus, mitochondria with double membrane, and chloroplast with double membrane. So here, prokaryotic cell, in order to evolve into eukaryotic cell, it has to produce or form all these cell organelles. So ribosomes, they are already present here. So let us skip the ribosomes. Then what about these? How they evolved? Scientists proposed two suggestions. One is that they must be formed by mutation. Mutation cannot give rise to all these. The second one, which more suits, is invagination theory. So what the scientists say is, if this is a prokaryotic cell, okay, the prokaryotic cell with a circular chromosome, single chromosome is present and 70S ribosomes. They say that the plasma membrane of this prokaryotic cell now has invaginated. So what happens soon? The invaginated portion gets separated, and this invaginated portion now becomes a single membrane bound cell organelles. So how the single membrane bound cell organelles like endoplasmic reticulum, Golgi complex, lysosomes are evolved? They evolved by the process of invagination of the plasma membrane of the prokaryotic cell. That is the first thing. Second one is symbiotic theory. Who proposed it? It is proposed by Margulis in 1970. So according to symbiosis means there are two organisms which benefit each other, which does not harm each other. So let us say
Yes.
there is any doubt?
No, sir. No, sir. Mutual benefit also.
Mutual benefit. Good. So there is a prokaryote, and a small prokaryote came nearby. Now what happens? This prokaryote, which is large in size, now engulfs the process which we call it as endocytosis or phagocytosis. It engulfs it. Now what happens? During, after it engulfs, it forms a structure like this. That is the membrane of this prokaryote, inside which this is the prokaryote with its DNA. So this is the prokaryote. Now this internalized prokaryote now is having two membranes. The membrane is the membrane of the prokaryote which allowed it inside, and the membrane of another prokaryote which is symbiotically living inside. So it is neither harming, because let us say this particular prokaryote is having chlorophyll, chlorobium chlorophyll. Let's say it is a cyanobacterium. So it can synthesize its own food material. So this prokaryote, which is said to be heterotrophic, now started depending on this, and this resides inside. And this particular prokaryote is given shelter, and this particular prokaryote is getting food, and both are said to be mutually benefited as suggested by you. And this gave rise to a double membrane cell organelle like mitochondria, chloroplast, and nucleus. Okay, and scientists wanted to prove this, so they have studied the mitochondrion. So the mitochondrion, when they have studied, the mitochondria is found to contain its own ribosomes. So a prokaryotic cell which is living symbiotically is having its own membrane surrounded by now the membrane of the original prokaryote which allowed it inside, and its own chromosome and its ribosomes. Likewise, mitochondria is it's also having its own DNA, its own ribosomes, and its own membrane, this one, and the membrane of the prokaryote allowed it. Now they wanted to see whether this membrane is having any relation to the membrane of the eukaryote, and this membrane whether it is having any relationship to the membrane of prokaryote. They have seen that the membrane organization of this mitochondrion is somewhat similar to the prokaryotic cell. In mitochondria, scientists say that, let us say protein synthesis. Protein synthesis is inhibited by two different chemicals. One is cycloheximide, the other is chloramphenicol. Chloramphenicol inhibits protein synthesis in prokaryotes, and cycloheximide inhibits protein synthesis in eukaryotes. When they have applied cycloheximide to mitochondria, protein synthesis is not inhibited. When they have applied chloramphenicol to prokaryotes, to the mitochondria, the protein synthesis in mitochondria is inhibited, suggesting that this prokaryote, this mitochondrion, this cell organelle is derived due to symbiotic origin. Thus, one by invagination. 2. By symbiotic association, prokaryotes gave rise to eukaryotes. This is what the theory of biochemical origin of life. Any doubt up to this point? You please ask me. Otherwise, we'll move ahead. Any doubt?
No, sir.
Okay. Now, let me quickly go through the other theories of origin of life. One theory is genes first hypothesis. Genes first hypothesis. That means one possibility is that the first life forms were self-replicating nucleic acids only, either RNA or DNA. So after only the lipid membrane around it is formed. We know that RNA can replicate, DNA can replicate. What is the first criterion for a protocell model? It should be self-replicating. So scientists say that genes means DNA and RNA might have first evolved, which are self-replicating. Hence, this is called as genes first hypothesis. Okay, and among these two, RNA and DNA, which one is first? That is what the question is. Scientists say in the first, when the life is originated, it is RNA that is present in the world rather than DNA. And this is called as RNA world hypothesis. It is proposed by Carl Weiss, Francis Crick, and Leslie Orgel in 1960s. So you have to write it down. RNA world hypothesis was proposed by Carl Weiss, Francis Crick, and Leslie Orgel. Okay, from which DNA later evolved. Why RNA only? DNA is not having any catalytic property. DNA cannot act as an enzyme, but we know that RNA can act as an enzyme, exemplified in today's world by ribozymes. So, if RNA, if any metabolic reaction to take place, it requires an enzyme. If DNA is formed first, because DNA cannot act as an enzyme, it cannot self-replicate itself first because it cannot act as an enzyme. So RNA evolved first because they have catalytic property, which eventually might have led to the evolution of DNA later. Okay. Next came metabolism first hypothesis. They say that self-sustaining networks of metabolic reactions may have been the first simple life. Okay, that means metabolism, without metabolism there is no breakdown of substances, there is no uh, release of energy. So they suggest that rather than genes first, metabolism first protocell models might have been evolved. And we know the theory of evolution of life. There are so many. You can, I will just run through them. Thales suggested that life originated from sea. So we call this as theory of aquatic origin of life or theory of marine origin of life. And somebody called as Anaximander. All these are Greek scientists. We can call them as Greek philosophers also. And origin of life took place from primordial fluid. That is what we say, hot dilute soup also. And theory of abiogenesis. Anaxi means air as source of life. It is also proposed by Aristotle later. And Xenophanes is another scientist who suggested that fossils are remnants of organisms living in past. And uh, we move to the most important things. The first one we start with Aristotle. Aristotle support the theory of abiogenesis proposed by Anaximenes, and he is called as a vitalist. What does vitalist mean? So he believes in the fact that though the organisms grow, reproduce, there is some inherent vital force which is responsible for all these activities. Hence, these are called as vitalists, and Aristotle is first among them. And he proposed a ladder of life, where he arranged all animals with increasing complexity. Simplest stars are to be placed down the ladder, and highly complex are said to be placed up the ladder, and those that are said to be in between are placed between these these two in the ladder. So this is called as ladder of life. So a question may come on this. Who proposed the ladder of life? It is nothing but Aristotle. Next came Jan Swammerdam. While we are discussing embryology, I proposed this preformation theory. That means the organism is said to be present either in the sperm or in the ovum. They are called as respectively spermis or ovis. And one scientist called as Hartsocker, he proposed a model where the human being is found to be sitting within the head of the sperm. He called this little human being as homunculus. That is what preformation theory is, and these need not needed for examination. And now there are certain other theories. One is called as encasement theory proposed by Bonnet. Write down what is encasement theory. For example, if somebody wants to give you some gift, and this gift is like this, they have packed it like this, that the gift is inside a small box which is wrapped by another box, another box, another box, and finally another box. Likewise, Bonnet suggests that miniatures of successive generations are said to be pre-existing one inside the other in the germ cells of the organisms. For example, in my germ cells, there are what happens in my germ cells? There are my children's miniatures. Okay, and in that there is next generation. For example, I am going to give rise to 10 generations. After 10 generations it is stopped, and I am having all the generations within my germ cells. And next, when I give birth to my children, the first box is opened and they came out, and they are having the remaining 9 generations inside, and the second box opened, my grandchildren will come. That is what Bonnet's encasement theory. It is encased. Okay, the second one is James Hutton, who proposed.
Sir,
Hello.
sir, సార్ ఒక ట్రీ తీసుకుంటే కింద కట్ చేసినాం అనుకోండి రింగ్స్ వస్తాయి కదా దాన్ని బట్టి మనం ఏజ్ చెప్తాం
Hmm.
అలా
అవును.
చెప్పారండి
And this body, any all these are we actually they're not theories, but they are not theories, they are hypotheses. వాళ్ళు ఊహల్లో తెచ్చినవి, వాళ్ళు ఊహల్లో తెచ్చిన వాటిని వాళ్ళు చెప్పేశారు అనమాట. ఇలా జరుగుంటుందేమో, ఇలా జరుగుంటుందేమో అని. సో అలాంటి థియరీయే ఇది. నెక్స్ట్ వన్ జేమ్స్ హటన్, థియరీ ఆఫ్ యూనిఫార్మిటారిanism. What does it mean? Life starting from Earth originated or life originated till now it is uniform. There is no change at all, according to them. And before Lamarck proposed, there is a scientist called as Buffon who proposed inheritance of acquired characters. We know that inheritance of acquired characters is proposed by Lamarck, but before that, a scientist called as Buffon proposed it. And later came the grandfather of Charles Darwin, Erasmus Darwin. So he wrote a book called as Zoonomia. Write it down. The book written by Erasmus Darwin is Zoonomia. Okay. It also gives support to inheritance of acquired characters, and he suggested the important concepts that were established later by Charles Darwin as origin of life by natural selection. What are those two important concepts? Struggle for existence and differential reproduction. These were first initially proposed by Erasmus Darwin. And next came the most important of evolutionists, that is Jean-Baptiste de Lamarck, who proposed the inheritance of acquired characters. We will deal with later. And after that, Cuvier, he proposed theory of catastrophism. So what does catastrophe? Catastrophe is a natural calamity, tsunami, volcanic eruption, and uh, glaciation, melting of glaciers. All these are said to be catastrophes. What he suggests is a big catastrophe led to the wiping out of all the creatures on earth. And life originated starting from this. Again, life went on uh, to produce complex organisms. Again, another catastrophe came, all these are wiped off. So, thus, successive catastrophes periodically destroyed all living beings, followed by successive creation of new and higher forms. That is what Cuvier suggests by his theory of catastrophism. Whether it is true or not, there is geological time scale which divides the entire time scale of origin of life till now into six eras. One is called as Ajoic era, followed by Proterozoic era, next Paleozoic era, next oh, I missed one. Mm, what is the one?
Mesozoic.
No. Ajoic, Proterozoic, uh, Paleozoic. I missed one thing. So followed by Mesozoic and Cenozoic. Cenozoic is I said to be the last one. So what I want to mention here is they suggest that after Ajoic era there is no life here. So nothing happens. Here the first glaciation might have taken place and entire earth is said to be washed off. And life originated and it gave rise to first life. That is Proterozoic era. Proterozoic era. That is what, and that led to Proterozoic era, that led to Paleozoic era, and finally Mesozoic and Cenozoic eras. In every, they suggest that every era is said to be wiped off by glaciation. There is a movie called as Ice Age, cartoon movie. So in Ice Age, what happens? They show us about the melting of glaciers, and all the organisms are wiped off from the surface of the earth, and life originates again from scratch. So that is what the theory of catastrophism is. And before this, James Hutton proposed theory of uniformitarianism, which was again supported by another scientist called as Charles Lyell. And Charles Lyell's hypothesis is also one of the what we call uh, evidences for Charles Darwin's theory. Okay, and he suggests that some organisms survived the catastrophe and gave rise to new forms, like them that is shown in the Ice Age movie. And later came Ernst Haeckel. He proposed two theories. One we have already discussed in embryology, that is life or recapitulation theory or biogenetic law, which means ontogeny repeats phylogeny. And the second one is theory of orthogenesis. You please remember this one also. Ernst Haeckel proposed two theories, recapitulation theory and theory of orthogenesis. According to orthogenesis theory, variations occur along certain guidelines, but they are guided by some inherent mystical force, as suggested by Aristotle. Here also, there is some mystical inherent force that guides the variations leading to the evolution of complex organisms according to orthogenesis theory. And next, there comes a theory called as modern theory of sexual selection, proposed by Julian Huxley. And Stebbins is the one who proposed the role of variations and hybridization in evolution in plants. And most important in the current days are these theories. The theory of natural selection, which is proposed by Darwin and simultaneously by Alfred Russel Wallace. And these two scientists proposed in separate countries. Later, they came to know that both are said to be one and the same, and they have formulated this theory that is called as origin of species by natural selection. But we attribute only this to Darwin. Why? Because both have proposed, but when the scientists were asking questions in seminars, only Darwin was defending. Wallace never might have never went for these conferences. So in due course of time, everyone is believing that only Charles Darwin proposed this theory. Okay, And later came Augustus Weismann's theory of germplasm. Lamarck proposed that uh, use and disuse theory, where somatoplasm is said to be responsible. Any change in the somatoplasm leads to the development of new organs, which are useful. And those that are said to be not useful, they are said to be completely eliminated. And that is what Lamarck's theory is. And this Lamarck's theory got a big blow when Augustus Weismann proposed that any change in the somatoplasm is not heritable. Any change in the germ plasm, that means in the reproductive cells, only can be transmitted to the next generation. Thus, Lamarckian theory is discarded by this theory. And later came Hugo de Vries, who proposed mutation theory. Mutation theory was a big blow to the natural selection theory of Darwin. Darwin believes in continuous variations. Darwin believes in continuous variations. What is continuous variation? Variations we know among human beings, we have so many characteristics. With respect to skin color only, there are so many characters, there are so many differences, and we call them as variations. So, for evolution to occur, a population should show a number of variations. According to Darwin, for evolution to occur, there must be continuous variations. What does continuous variation? Let me give a small example which is not there in the books. It's just for your understanding. There is a small lizard. Okay, lizard is very small, from which they evolved the dinosaurs. Why I'm saying this example? We call lizards in Telugu as Balli and dinosaur as Rakasi Balli. Okay, so what according to Darwin, the variations are continuous. That means this gave rise to somewhat a larger sized one, somewhat a larger sized one. Ultimately, it gave rise to the dinosaurs. That means in fossils, we should come across with all these forms so that the variations are continuous. It is
Sir,
saying,
deer and giraffe.
uh,
Deer and giraffe, sir. Like,
likewise,
like
so many
smaller.
examples. Just for understanding only these are required. So now, mutation theory was proposed by Hugo de Vries, who, according to which these are resulting in discontinuous variations. So mutations are considered as the raw materials for evolution. That means discontinuous variations are said to be causing evolution, which got a big blow to the theory of natural selection. After that, Wenger proposed isolation theory. Yesterday we have discussed that geographical isolation is required for the formation of new species. New species. Without new species, there is no evolution at all. And for new species to occur, first they have to be isolated to the new environment, which is called as geographical isolation. Not only geographical isolation, these are so many isolating mechanisms proposed by Wenger. And later came Sewall Wright's genetic drift theory. It is also called as decay of variability. Decay of variability. It is also called as scattering of variability. Genetic drift is having different names. Decay of variability or scattering of variability. What is genetic drift? It is evolution in small populations. In simple terms, it is evolution in small populations by chance fluctuations. By chance fluctuations. Okay. In a box, there is a red ball and a white ball. Okay. When you pick up a red ball, when you were asked to pick up one ball, you can pick a red ball or white ball, and each one is having 50% probability because there are only two, but there are 100 balls which are of different colors. 100 balls of different colors, and I'm asking you to pick only one that should be red, and the chances are said to be the probability will decrease because the population size is increased. So when the population size increases, there is less chance of the new mutation to be fixed in the population. When the population size is said to be small, there is every chance for the new mutation to be fixed in the population. That is what genetic drift theory simply is. And coming to the patterns of evolution. There are different patterns of evolution: sequential evolution, divergent evolution, which is also called as adaptive divergence, convergent evolution, which is also called as adaptive convergence, parallel evolution, iterative evolution, concerted, monophyletic, and polyphyletic. Let us see one by one. First of all, uh, in this classification, after this classification, we come across with the definitions of all these. First of all, Goldsmith divided evolution into three types. One is called as microevolution, the other is called macroevolution, the third one is megaevolution. And Wrench is another scientist who divided evolution into three types: anagenesis, cladogenesis, stasigenesis. And George Gallord divided evolution into two types: bradytelic and tachytelic. And we are going to deal with all these different types of evolution. According to Goldsmith, three types of evolution: micro, macro, mega, three M's. Okay. And according to Wrench, it is anagenesis, cladogenesis, stasigenesis. And according to George Gallord, it is bradytelic evolution and tachytelic evolution. So, what is anagenesis? A species slowly accumulates changes until it reaches a point where it is no longer the same species it was thousands or millions of years ago, according to anagenesis. That means there is species A which was slowly accumulating changes. Some changes in its skin color first, followed by its hair pattern, followed by its color of the eye, followed by internal organs, external organs, hands, feet, limbs, etc., etc., etc. In due course of time, what happened is, after millions of years, it was formed into one species which in no way resembles the ancestor. And such accumulation of changes leading to the evolution of a new species which is nowhere related to the ancestor is called as anagenesis. Second one is cladogenesis. Here one species splits up into two or more groups, with each one evolving on its own path until the groups are now different species. We know that species, Homo sapiens, is a species. Species is always divided into different types of populations. So a species contains a different types of populations. For example, human beings, American population, European population, Asian population, even in Asia, Chinese population, South Korean population, Indian population, even in India, North Indian, South Indian, even in South Indian, Andhra, Telangana, and so on. So a species is divided into small populations, and this population which lives in a special circumscribed or unit area, they live in a unit area. When the population census takes place, they measure how many individuals or human beings are present in Vishakhapatnam, how many are present in Kashmir, how many are present in uh, Kolkata. Likewise, so this is a unit area. In this unit area, there are so many individuals. All these individuals belong to the same species, but they are restricted to one area. They are restricted to one unit area, and these are not combining with the other population. So they exhibit interbreeding. They share a common gene pool, and such a group of population which are said to be what we call small enough that can interbreed among themselves, share a common gene pool, constitute a Mendelian population or a deme. Okay, so here species is divided into small populations. Let us say there is a species with a number of populations living in this area, and one population went ahead into crossing a large mountain. Okay, it went on to the other side. Okay, because why it has happened? Because there is a competition among these organisms. As they are growing in number, there is competition for food and also some other thing. So they are separated by a geographical isolation. So it got into split into one population. So still too, some of them crossed a big river and they migrated to different locality. So thus the same pop species is divided into small groups, and the ones that are living here after this to be prone to a different set of environmental conditions. The ones that are present here, they are prone to different set of environmental conditions. For example, these are said to be present in like Himalayan region. So the environment is completely different. The organism has to adapt itself to the newly changed environment where it has to accumulate fur on its body. Okay, if some goes to Arctica and Arctic or Antarctic regions, what happens? They have to develop a complete polar bears like thing, isn't it? Polar bears. There is a normal bear here. There is a polar bear here. Polar bear is said to be withstanding the temperature. So it has certain new environmental features for which it has to adapt itself. And when this happens, it results in the formation of new species, which is called as allopatric speciation. What happens here? One species splits into two or more groups, each one evolving in its own path until the groups are now different species. In such case, it is called as cladogenesis. Next one is stasigenesis. Here, the lineages, they never split, they never change, but they remain unchanged. We know there are coelacanth fish. Coelacanth fish is dating back to 17 million years ago. Its ancestors are present 17 million years ago, where they are present in the South African rivers. The same fish are present in South African rivers. From where? 17 million years ago. And when the scientists found the ancestors of coelacanth, there are the structural and functional similarities are so many between these two. That means what happened here? The coelacanths remained unchanged for about 78 million years. How does it happen? Only change in the organism occurs only when there is a need. How the need occurs? When there is a change in the environment. Here, the South African rivers, let us say, all through these years, the environment did not change at all, and the coelacanths does not require any change at all. So they remained unchanged, and there is no evolution at all here. And such an evolution is called by scientists as stasigenesis. Likewise, turtles, we also call them as living fossils. Pinodon, we call them as living fossil. Coelacanths, we call them as living fossil. All living fossils exhibit stasigenesis. And next one is brachytelic evolution, which is also called as gradualism. So here gradualism means evolution of new species occurs slowly. There are two terms, bradycardia and tachycardia. Cardia means heart. Brady means slow heart rate. Tachycardia means faster heart rate. Likewise here evolution also there is bradytelic and tachytelic. In bradytelic evolution, the evolution of new species is slow and gradually. So exemplified by living fossils as in the case of stasigenesis and evolution of cockroaches, termites. And tachytelic evolution, here it is rapid, it is faster. Origin of species takes place by abrupt changes. So abrupt changes means a lizard giving rise directly to dinosaur. There are no intermediate forms in between. And when intermediate fossils forms are absent, that means the evolution is occurring at a rapid rate. Then such an evolution is called as tachytelic evolution. It is also called by scientists as punctuationism. Example, evolution of amphibians, evolution of reptiles, and evolution of birds. So tachytelic evolution is called as punctuationism, which you have to remember, and bradytelic evolution is called as gradualism. And microevolution. Yesterday we have discussed about it. What is microevolution? Formation of subspecies only. Macroevolution. It is nothing but formation of new species which are adapted to the newly changed environment. And macroevolution also refers to as adaptive radiation. What is adaptive radiation? Ancestors are present at one place and this population is split like in cladogenesis into small subpopulations. They went to different areas. For example, mammalian ancestors. All these are present in the same area, mammalian ancestors, and all are having same characteristics. But competition arose, They have to go to different areas and they started migrating. And some of them reached what we call forest area. Some of them reached forest area. Some of them went to aquatic area. Some of them went to hilly areas. Some of them went to plain lands. Now what happened? During the course of time, they evolved into forest dwelling mammals, aquatic mammals, and what we call cursorial mammals, fossorial mammals, and so on. Different types of cursorial means horse-like things. Okay, and cave dwelling animals like bat-like things. So all these arose from a common ancestor. There is a sun from which the light rays radiate in different directions. Here there is an ancestor from which the populations radiate into different directions and evolve into different species. And hence we call it as adaptive radiation because they have to adapt themselves to the newly changed environment. And coming to the mega evolution gives rise to the formation of classes, groups, or phyla. For example, can anybody tell me what is the connecting link between reptiles and birds? Connecting link
Archaeopteryx.
Archaeopteryx. So evolution of Archaeopteryx led to the evolution of birds, that is aves. This is reptilia. Reptilia and aves, are they classes or phyla or orders?
Classes,
Reptilia
sir.
class. So what happened? Evolution of Archaeopteryx led to the evolution of a new class, that is aves. So when such evolution takes place, evolution of new formation of new class, we call it as mega evolution. Likewise, what is the connecting link between annelids and arthropods? Annelids and arthropods.
Catopteryx.
Peripatus, let us say so, which was later separated to form one Icophora. It is having both annelid and arthropodal characters. So Annelida is a phylum, Arthropoda is a phylum. So evolution of this particular organism led to the evolution of a phylum, and such evolution is called as mega evolution. Origin of reptiles from amphibians, origin of birds and mammals from reptiles. Classes evolved here, and then we call them this as mega evolution. Coming to the population genetics, gene pool we start with. Sum total of genes present in a Mendelian population is called as gene pool. What is Mendelian population? The population which is present in a unit area that is sexually interbreeding and shares a common gene pool. Okay, so all the genes present in this Mendelian population constitute gene pool. And the gene pool, when we refer, we do not refer to diploid condition, we refer to only haploid. That means first chromosome is a pair. Whatever the genes that are present here, the same genes are present here also. When we consider these two, we need not consider these two. We have to consider only one. Thus, we consider only 23 chromosome for counting the sum total of genes. So, thus, these are present in haploid gametes. So, gene pool is also referred to the gametic pool. Okay, and the genes, for example, there is first chromosome on first locus. We are concerned with gene responsible for eye color. Okay, so let us say there is a gene for black color, capital B. There is a gene for brown color, small b. So there are two genes. There are 100 individuals, and if in this 100 individuals, 50 are said to be black and 50 are said to be having brown eyes. What is the frequency? 50 by 100 and 50 by 100. That is 0.5, 0.5. Combined with these two, it is 1. So in one, half are said to be black colored, half individuals are having black colored eyes, and the other half are said to be having brown colored eyes, and the frequency is said to be 0.5 for each. So we have to count the gene frequency. So what happens according to Hardy Weinberger when selection, mutation, and gene flow occur? So 50 are having black eyes. If 5 of them got migrated to some other place, the frequency now decreases to 45 by 100. So the frequency, gene frequency decreases when there is mutation, when there is migration, gene flow. And if about 5 are mutated to form small b, what happens? The small b frequency increases, capital B frequency decreases. So even if mutation occurs, there is a change in the frequency. If the black individuals are marrying only black and also brown individuals, but the brown individuals are marrying only among themselves, what happens? The brown individual frequency decreases and the black individual's frequency increases. If there is a selection, also there is a gene frequency change. So selection, mutation, and gene flow are the ones which bring about a change in the gene frequency. What do you mean by gene frequency? Previously there is only black gene and a brown gene. Now it got mutated to form a blue gene. Now it got mutated to form a and the green gene. So here there are variations being produced, new characters being produced in the populations because of new combinations or recombinations. So gene frequency shows that the population is in the path of evolution. New characters are accumulating the population. In due course of time, it may lead to the formation of a subspecies. In due course of time, if chromosome mutations occur and reproductive isolation sets in, a new population, a new species may originate, and we say that evolution has taken place. So gene frequency is one of the criteria to measure what we call evolution. So the gene pool has certain characteristics. It is integrity. That means the all the individuals, human beings, are having the same gene pool. They are not sharing with other ones. And the gene flow, they can migrate so that they can carry the genes away from this. Equilibrium, all the genes in a particular population exhibit equilibrium unless otherwise there occurs a selection, mutation, or gene flow. And there are fluctuations in size of gene pool due to mortality or again selection, mutation, or gene flow. And how we calculate gene frequency? So it is defined as the proportion of an allele in the gene pool. Here there is a capital B, small b. Proportion means there are 100 genes total. How many? 50 are said to be, let us say 60 are said to be capital B, 40 are small b. That means 60 by 100, 0.6 is its gene frequency. The proportion of an allele in the gene pool as compared with other alleles, on the same locus is called as gene frequency. And gene frequency calculation, how it is done? Let's take three types of individuals: homozygous dominant, capital A, which are 25%; heterozygous dominant, capital A, small a, 50%; homozygous recessive, small a, 25%. And here we are concerned with only one locus. Few individuals are like this, and another individual with the same locus on the same chromosome are like this, and another individual on the same chromosome with the same locus are like this. And the proportion is 25%, 50%, and 25% respectively. And then now we have to calculate the total number of individuals. So total number of individuals, let us say all these are 100. So n is 100 here. Total number of alleles A and A becomes 2n. So one person with this locus, how many are present? 2 loci. So individuals are 100, but each individual is having 2 loci. That means 2 genes present. And now all the 100 individuals will have on the same locus how many? 200 genes. So the total number of genes or alleles in a diploid individual is 2n in that particular population. Now the number of homozygous dominants is denoted with capital D, and recessives with capital R, and heterozygotes with capital H. Okay, and now the total number of dominant alleles, the total number of dominant alleles. That means they are homozygous dominants. In this also there are dominant alleles, and in this also there are dominant alleles. So the total number of dominant alleles is 2D, means number of homozygous dominants. So here there are two genes. So this is dominant gene, there another gene. Thus it is 2D plus here in heterozygote also there is a dominant gene. So 2D plus H is the total number of dominant alleles. So total number of dominant alleles contributed to the gene pool by homozygous dominance is 2D, A plus A means D plus D, 2D. Number of dominant alleles contributed to the gene pool by heterozygotes. That means capital A, small a, only one is contributed, not to H. We call it as only H. H. Thus, frequency of gene A is 2D plus H by total number of genes present in that population. Population is said to be 100. Total genes are said to be 2 into 100. Here n refers to 100. Thus, it is 2D plus H by 2n. Likewise, for recessive gene, the gene frequency is 2R plus H by 2n. Here 2R refers to small a and H refers to the A gene contributed by the heterozygote and 2n refers to the total genes present in the population. This is how you have to calculate gene frequency. And according to Hardy-Weinberg law, the dominant gene is always having a frequency of small p and recessive gene is having a gene frequency of small q. In the yesterday's experiment, we have seen that 36% of population are said to be having a homozygous dominant genotype, even in the parent generation as well as in the children generation, progeny generation, first generation of children. And here this frequency is small p and this frequency is small p. P into p is equal to p square. And the homozygotes are 48%, isn't it? And here how 48% arose, 24% are capital A small a, 24% are capital A small a. That means PQ plus PQ, that is 2PQ. And 16% are small a and take Q into Q, that is Q square. So when you combine all these, they are said to be counting to 100 and 100 can be taken as unit 1 or it can be taken as 1000, so on, depending on the number of population. If the population size is 100 and that population size is considered as unit 1, what happens? P square, that is 36 plus 2PQ, that is 48 plus Q square, that is 16 is equal to 100. If this is unit 1, P plus Q plus U whole square is equal to 1. Because the population size need not be always 100, it can be 200, it can be 1000 and we are counting it as 1. So by reducing it, we can call it as P plus Q whole square is equal to 1. That is what Hardy-Weinberg's law is. And coming to the genotype frequency. So far we have calculated the number of genes, capital A or recessive genes, dominant 50 by 100. That means 1 by 2, that is 0.5 is its gene frequency. And small a, they are 25%. 25 by 100 means their gene frequency is 0.25. So 0.25 plus 0.5 plus 0.25, ultimately it is also coming about 1 only. So in genotype frequency, we measure only the frequency of homo or heterozygotes. Whereas in gene frequency, we measure only the genes, not the individuals here. Okay. And Hardy-Weinberg law, we already have discussed about it, and I'm not going to that again. So what in Hardy-Weinberg law, what you have to remember is random mating, sexually reproducing population, which is large in size, and there must be absence of selection, mutation must be absent, gene flow must be absent. When all these 6 criteria meet, what the scientist says, gene frequency remains constant. What does it mean? There is no evolution at all. So he suggested a hypothetical situation where there is no evolution. Opposite to this, when selection is there, mutation is there, gene flow is there, gene frequency changes in small population which is mating random, not randomly, which is exhibiting selection. In that case, there is evolution happening. So Hardy-Weinberg law tells us about a hypothetical situation where there is no evolution at all. In other words, what are the conditions for evolution to occur is explained indirectly by Hardy-Weinberg law. And coming to the forces of evolution, there are so many forces, and one force is said to be variations. Variations are nothing but changes in all morphological and physiological characteristics like shape. The shape of the nose is not same in all human beings. There is a variation. Size, height is not same, and color is also not same, and so many characteristics, physiological characteristics with respect to organ systems. The shape of the liver may change, and also so many characteristics. And the mental ability, IQ, is also different. All these all these are called as variations. There are so many types of variations, and among these, in the examinations, these two only will be asked. Continuous variations or fluctuation variations, which we have already discussed while dealing with Charles Darwin's evolutionary theory. He suggests that continuous variations are raw materials for evolution, according to natural selection theory, which is not true. Discontinuous variations or mutations are responsible for evolution to take place. And variations which occur in somatic cells are not heritable. They are called as somatogenic variations. And those that occur in germinal cells, reproductive cells, only are heritable. And they are called as germinal variations. And now we move to mutation theory proposed by Hugo de Vries. Mutations are nothing but sudden heritable changes in the genetic makeup of an individual which are heritable, and these are the ones which are called as the raw materials for evolution. So if any question is asked like what are the which of the following are said to be raw materials for evolution, you have to uh, tick the mutations as the correct one. And discovery of mutations. De Vries discovered mutations while working on Enothera lamarckiana, which is commonly called as evening primrose. He observed different category, about 8 varieties he observed it. And the one question that may be asked in this part is, the first one, on which plant or organism he worked on. What is its common name? And what is the name of the theory? Who is the scientist? And the fifth one that is generally asked is, what is the mutation that was observed by De Vries in Enothera? And you have to answer it. It is the translocation mutation. That is the mutation that is observed by Hugo De Vries is translocation mutation. Okay, and let's move to mutagens. Mutagens. What is a mutagen? Any agent or any substance that causes mutation is called as a mutagen. And radiations are also mutagens. Chemical substances are also acting as mutagens. So coming to radiations, X-rays, alpha, beta, gamma, ionizing radiations, neutrons, protons, UV rays, cosmic rays, and radioactive elements. All are mutagens. And likewise, coming to chemical mutagens, formaldehyde, mustard gas, caffeine that we take during tea and coffee, and phenol and organic agents. All these are said to be chemical agents. And mutations cause so many types of diseases which we have already discussed while we are dealing with mutations class. So another aspect I want to mention is polymorphism. What is polymorphism? Existence of two or more forms of the same species within the same population at the same time and place is called as polymorphism. For example, we have hemoglobin. Hemoglobin is fetal hemoglobin present in the fetus, adult hemoglobin present in us, sickle cell hemoglobin present in the sickle cell anemia individuals. So, in a given population, we can see all these three present on the same population. and such in the same species of the population, and hence we call this as polymorphism. Okay, and there are two types of polymorphism: balanced polymorphism and transient polymorphism. In examination they will be asking about balanced polymorphism. So what is balanced polymorphism? We can explain it by the example of what we call sickle cell anemia. For example, in when we take Plasmodium vivax, it causes malaria. Plasmodium vivax lives in RBC and also liver cells. So its food is said to be hemoglobin. It eats hemoglobin. Okay. So its residence is RBC, which are biconcave. Now what happened is sickle cell anemia occurred. The biconcave RBC became sickle shaped. Because it became sickle shaped, the Plasmodium vivax is not able to live here because the sickle cells are said to be having more surface area. As the blood flows, they break, they become fragile and they break. There is no hemoglobin for the Plasmodium vivax to eat, and it cannot survive there. And that is the reason why generally sickle cell anemia is fatal. Individual which is having this gene will die, but there is a probability that there is a normal gene and a sickle cell gene. This is a heterozygote individual, and this heterozygote individual is having an advantage in malaria because 50% of the blood is sickle cell, 50% is normal, and this is said to be not harboring malarial parasite. So these individuals were resistant to malaria. So in this population where there is malaria prevalent in certain agency areas, this gene is maintained. Generally, when this gene is said to be homozygous recessive, the individual dies and from the population the gene is said to be eliminated. But here this is said to be having a heterozygote advantage. We call it as heterozygote advantage. In recessive, it dies. In normal, it survives. Even in heterozygotes, it is surviving because it is having an advantage. And individuals with two or more forms coexisting the same population of a species in stable environment and showing almost constant ratio is called as balanced polymorphism, exemplified by sickle cell anemia, ABO blood groups, and others. So, in general, it is asked about sickle cell anemia. And let's move to isolation is not required here. And I want to mention something about natural selection and the process called as differential reproduction. So, for evolution to occur, one of the features is differential reproduction. What is different? Sometimes this question may be asked. What is differential reproduction? It is the differential contribution of offsprings to the next generation. That is, individuals that are best adapted to the environment reproduce at a higher rate and produce more offsprings than those which are less adapted. For example, there are two types of individuals. So these are adapted to the environment more than this one. So they reproduce and produce more number of gametes so that more progeny are said to be produced. And when compared with this, these are less adapted, they produce less number of gametes and they produce less number of individuals. In such case, there are two types of individuals exhibiting different types of reproductive capability, and we call this as differential reproduction, and nature selects these individuals rather than these individuals. So nature selection goes for a property called as differential reproduction. There are three types of natural selection with which I will end this class: directional selection, stabilizing selection, and disruptive selection. Directional selection is exemplified by industrial melanism. Industrial melanism. What is industrial melanism? Previously, before industrial revolution has taken place in
Yes.
England, the barks of the trees are said to be
Bark.
white. They are said to be white because of snow. And the peppered moth, distant Beetleria, is said to be two types. One is said to be grey in color, the other is said to be black in color. So when the black one comes to occupy a place on the barks of the trees, it is easily recognized by the birds and it is eaten away. So the grey population increases and black population decreases. After what happened, industrial revolution taken place and the barks of the trees are said to be occupied by the smoke. And now there comes a black one and also a white one. And now this white one is said to be, the grey colored one is said to be clearly visible for the predators and they eat away it. And thus, before industrial evolution, there is one species that is said to be predominant, that is black one. After industrial evolution, it is one grey one. After industrial evolution, it is the black one that is predominant. That means in a curve, when you present it in the form of a graph, you see only one population at one time which is replaced by another one at another time. So there is evolution occurring in a direction. Towards the head end, this is tail end. Tail end, those that are present towards the tail end, they are eliminated, and towards the head end, they are said to be selected. Thus, you can see at a given time only one kind of population here. This is what we call it as direction selection. Second one is stabilizing selection. When we consider the newborn babies, the newborn babies can be uh, 1 to 2 kgs in weight. There can be 3 to 4 kgs in weight, and they can be 0.15 to 1 kg in weight. In all these, if they are 0.5 to 1 kg in weight, they cannot survive. If they are 3 to 5 kgs in weight, the mother cannot bear them. If they are 1 to 2 kgs in weight, it can be they can give birth. The mother can bear them, and the babies can also survive. So here in a graph, when you see, these are extremes, this is extreme and this is extreme, and only the mean character is selected. And in the population, when you see every child that is born in that population, if it is having only 1 to 2 kgs, we say that this character is said to be stabilized, and such a selection is called as stabilizing selection. And the third one is disruptive selection. For example, let us take the height characteristic. In height characteristic, there are dwarfs, there are long individuals. Both are present in the extremes, and also different kinds from 3 feet to 4 feet to 5 feet to 6 feet to 6.6. All these are present. That means the extremes are selected and the mean is also selected. That means different variations are present in the population, and such a selection is called as disruptive selection. Among these, the population that is having variations is that shows disruptive selection, and this is the one which is important for evolution rather than these two. So these are the few things I want to uh, give a brief insight before tomorrow's examination with respect to evolution. When we start the next phase of classes, I will be dealing with each and everything in detail. So thank you for watching all these, joining all these days for the classes. For the tomorrow examination, who are writing life sciences, all the best. God bless you. బాగా చదివింది ఏదైతే ఉందో మంచి క్వశ్చన్ ఒకటి రెండు సార్లు చూసి ఆన్సర్ చేయండి. ముందు వచ్చినవన్నీ ముందు పెట్టేయండి, రానివన్నీ లాస్ట్లో ఉంచుకోండి. వాటిని ఆలోచించి పెట్టడానికి ప్రయత్నించండి. ఓకే, ఆల్ ది బెస్ట్.